Excellent. Okay. So, so again, thank you so much for being here. If you want to see any of the details of some of the biology that I'm going to show you, please don't hesitate, get in touch with me. There are lots of protocols, papers that have all the details and kind of all the nitty gritty. But what I'd like to do is share what I think are some of the key messages from the work that we've been doing. And you can find me at these two sites. So, the main points for today, I want to tell you a few things. I want to tell you that biological systems have remarkable structural and functional plasticity that goes really way beyond the default outcomes that we associate with any particular species. And I want to introduce you to the concepts of anatomical homeostasis and the idea that there is some top-down control of collective outcomes and that it's not all emergence from the bottom up. And I want to tell you that bioelectric networks is how evolution expanded the boundaries of cells into organisms, allowing their goals to get bigger. And this has really important implications for understanding the reprogrammability of life and the hardware-software distinction that I'll try to make about living things. And the hypothesis that I want to put out there is that in biology, there's this multi-scale autonomy of goal-seeking subunits, which brings the risk of cancer, and it brings the potential for some of these subunits to defect from the global plan. But actually, overall, is the key to adaptive robustness, and it's what allows biology to be extremely reliable at the large scale, despite the fact that it's working with huge amounts of noise and unreliability at the microscale. And we'll talk at the end about how I think this really facilitates evolvability in general and why life is so highly evolvable. So in a single sentence, what I'm going to show you is that, like the brain, somatic tissues form electrical networks that make decisions. And these are decisions about anatomy. And this is a proto-cognitive system that can be exploited to understand how we can program cell swarms top down towards large-scale outcomes. And I'm showing you this five-legged frog to warn you in advance. You're going to see all kinds of really weird creatures in this talk. None of this is Photoshop. These are actual living things that are in our lab. And we create them to test our theories of how cell collectives are guided towards specific anatomies. So this is at the center of a really interesting emerging field that intersects developmental biology, basal cognition, and computer science. What we see is that there is complex decision-making at all levels and scales, all the way from molecular networks up through groups of organisms and everything in between. And these parts are unreliable, but very smart. And the magic of biology appears through the combination of bottom-up emergence and top-down representation and reprogrammability. And I'm going to show you some examples of this. So let's start with this. This is a simple example of the robustness of function in altered body plans. What you're seeing here is a tadpole, and what you'll notice is that this tadpole of a frog, it is missing its primary eyes in the head, so there are no eyes in the head, but we've induced an eye to form in the tail. And I'll tell you later how we do this, but here's an eye in the tail. And the amazing thing is that this animal and this brain, which evolved for millions of years to expect visual input from a particular location in the head, these animals can actually see out of that eye perfectly well. And the reason they can see is because this eye, finding itself in the tail, puts out an optic nerve. The optic nerve looks around, it finds the spinal cord, it synapses on the spinal cord, and then that information gets passed up towards the brain. And so look at the incredible plasticity of this system. All of the behavioral repertoires, the visual learning and everything else, are remapped onto this novel architecture where the sense organs are in the wrong location. And so what this is showing you is that the brain can dynamically adjust its behavioral programs to accommodate different body architectures. But why would this be? I mean, most tadpoles are perfectly normal. The vast majority of them have the eyes in the correct spot. Why would this plasticity exist? And this is going to be a theme throughout the talk. So let's think about now the relationship of the mind, brain, and the body. Here on the left, you see a caterpillar. This is a soft-bodied robot. It crawls and it chews plants. This thing has to become something very different. It has to become a butterfly, which is a hard-bodied robot that flies and drinks nectar. The amazing thing is that the memories that caterpillars make survive into butterflies. And what's amazing about it is because during this process, during metamorphosis, as the caterpillar becomes the butterfly, its brain is basically liquefied. So most of the synapses are taken apart, many of the cells die, and it basically reassembles a brand new brain. And yet the memories remain. So what is this? What are the implications of this for models of learning, for example, in artificial neural networks, where all of the information during learning is stored as these very finely tuned synaptic weights? I mean, this in biology, all of this can be drastically remodeled and altered, and the information still remains. So we really need to understand how this works and how the biology manages to do this kind of transfer learning, in effect, of taking information that made sense to a caterpillar and converting it into information that's suitable for a completely different body architecture. Okay. Now, in planaria, these are flatworms that you'll hear a lot about today. This is even more profound, where we can take a single worm. We can teach them something simple, like to associate a certain pattern of roughness on the floor of the dish with their food. We then chop off the head and the brain. We take the tail fragment. The tails regenerate. That's one of the remarkable things about planaria, is that they regenerate entire bodies. They will regenerate their brain. And as soon as that brain is complete, the rest of the body has imprinted that information onto it, because now the resulting animal still remembers. Okay, so the memory is stored somewhere else. We're not exactly sure where, but it's imprinted onto a brand new brain. So these kind of models show you that in biology, we don't just deal with a static or a fixed type of organism, and we ask how it has behavior and memory and IQ and things like this, but really the body is incredibly plastic. And these types of models raise questions about what really is a memory. You know, how is it stored? How is it encoded? How is it decoded and transferred from one set of tissues onto another? And this has not only major biomedical implications, because of course, pretty soon we'll all be receiving transplants of naive stem cells for various degenerative diseases of the brain, but also it has major implications for philosophy of mind, and asking what exactly is a memory that it can be housed in a substrate like this that can be altered. Okay, so we really need to understand what controls the pattern of the body in the first place. And this, it may seem to a lot of people that biology kind of has this handled, but I want to show you that there are still deep puzzles in this area. So we all start life as a single cell, a fertilized egg, and this divides a few times, and we have these embryonic blastomeres, a bunch of cells sitting together. And then they reliably self-assemble into an incredibly complex machine. What you're seeing here is a cross-section through a human torso. So you see that all of these organs and tissues have to end up in exactly the right location, exactly the right topological relationship to each other. And so one question is, where does this pattern come from? And of course, immediately people think of DNA, but if you actually read a genome, what you see is protein sequences. You don't see anything in the genome directly about the type of symmetry that this animal is going to have, the size, the shape, the kinds of organs. None of that is directly in the genome. All the genome tells you about is proteins. So the question is, how do cell groups actually know what to make? How do they know when to stop? What can we do to convince them to repair this kind of structure if it's been injured? And of course, as engineers, and especially in synthetic biology and artificial life, we would like to know, how far can we push this process? So if we wanted these cells to build something completely different, okay, would that be possible? Or can they only make one thing? And I'm going to show you that the answer is they can build lots of different things. So the current paradigm for all of this is something like this, that the genes interact in networks. Some of these networks make proteins. The proteins interact via physics, so maybe they're sticky, or they exert force, or they diffuse, they do various things. And then, of course, there's this wonderful process of emergence, and out comes something amazing like this salamander, this axolotl. But this has a fundamental difficulty, and the difficulty is a kind of inverse problem. What molecules would you tweak at the lowest level of description to give you desired system level outcomes? And it's a very hard problem. It's a problem both for biomedicine, but also for evolution and for the animal itself. And the amazing thing, of course, is that embryogenesis is reliable. It's extremely reliable, but it is not hardwired. And so, for example, if you take an early embryo and you split it in half, you don't get two half embryos the way you would with most of our human artifacts, but you actually get two perfectly normal monozygotic twins. You can do the opposite experiment. You could take two mouse embryos, for example, mush them together like a snowball, and out comes a perfectly normal mouse. So what you're seeing here already is that while the process is reliable, it has the ability to regulate to a specific outcome following really drastic perturbations. Okay, so it's not hardwired in the sense of having the same set of steps each time. In adulthood, some animals like this axolotl are able to perform this trick, and they regenerate large portions of their body. So these axolotls can regrow their limbs, their eyes, their jaws, parts of the brain, their heart, tail, ovaries, and various other organs. And if you amputate the limb, what happens is it will undergo a set of deformations, a bunch of cells will grow, and then it makes a perfect replica of the original, and then it stops. And this is the most profound mystery out of all of this, is how does it know when to stop? Because it stops exactly when a correct salamander limb has been formed. So we really need to understand that process. And this is an old experiment that was done that illustrates the kind of the kind of analysis that the system is able to do, where what they've done is they took a tail from a salamander and they surgically grafted it onto the flank where where a leg is supposed to go. And what happens is within some amount of time, this tail remodels. It becomes a leg. Even the cells at the tip of that tail start to become fingers. So what you're seeing here is that the individual cells are given cues that are counteracting what their local situation is. Locally, the cells at the tip of that tail are fine. They're at the, they're at the end of a tail, so they're doing fine. But globally, this is not correct with respect to the overall structure. So there's this large-scale body plan, and there's information that's constantly making sure that the individual cells are doing the right thing to be a correct structure. So this theme of an anatomical set point comes back again and again. And in planaria, it's even more impressive, where so here are these flatworms. They have a brain, they have a nervous system, most of the same neurotransmitters that you and I have. And they have this amazing ability that you can cut them into pieces. In fact, any plane that you want. I've only shown one kind of cut here, but you can cut them in any direction. The record is something like 275 pieces, and every piece will regrow exactly what's missing, no more, no less, until you get a perfect little worm. So this is an amazing system that has this kind of holographic type of knowledge of what a correct worm looks like, because every piece of this can do it. And in fact, they're so good at it, they're basically immortal. There's no such thing as an old planarian. The individual cells will senesce and die, but it continuously regenerates them. Okay, so the animals that we have in our lab are basically in direct physical continuity with animals that were here almost 500 million years ago. So you can see the power of this ability to restore to a particular shape. And this is not just for so-called lower animals. Human liver, of course, regenerates. Children's fingertips regenerate from certain types of amputation wounds. And deer, a large adult mammal, also regenerates huge amounts of bone and innervation and vasculature and skin every year as they shed and regrow their antlers. Now, one of the cool things about regeneration and development really is that here you can see how large scale patterning information overrides individual cell states. So if you have a tumor on a limb and you amputate through the middle of that tumor, or you take aggressive carcinoma cells and you stick them into into a mouse embryo, you will get a perfectly normal mouse and you will get a perfectly normal salamander limb. These cells are being normalized by the information that's coming down towards them from the rest of the body, telling them what they should be doing. And this is really key. These large scale global patterning plans are really important. So what I've told you so far is that living anatomy is incredibly plastic, and it has the ability to repair toward specific anatomical states from different starting conditions. And this is I think the right way to think about this is as collective decision-making in cell swarms. Now, most of biology is currently focused on thinking about stem cells, the role of stem cells in this process, and genomic editing, and working at the level of the genome. And I'm going to show you at least two of the major puzzles that we have. And this is why I think fields like artificial life have potentially a huge amount of impact that they can make in regenerative medicine and biomedicine as a whole. So one question would be this. So planaria, unlike the rest of us, most of us, if we have a mutation in our body and during our lifetime. Our children do not inherit that mutation. But planaria, because some species reproduce by fission, meaning they just divide, they tear themselves in half, and each half regrows, and now you got two worms. So that means that they have what's called somatic inheritance. Every mutation that doesn't kill the stem cell ends up propagating and amplifying in the next generation. So their genomes are an incredible mess. Hundreds of millions of years of accumulating all these mutations. In fact, we don't even have a proper assembly for the species that we work with. The cells are mixoploid, meaning they have different numbers of chromosomes. So the genome is all over the place, but they are rock solid regenerators, 100% perfect every single time. And so it's quite clear that we don't really understand very well the relationship between the genome and the anatomy, when you can make such a mess of the genome and still have complete total anatomical fidelity. And in fact, there's a thought experiment you can do, which is that if you take two planaria with different head shapes, so you have let's say a flat head and a round head planaria, and you take some stem cells from the flat head and you inject them into the round head, and this is a procedure that people do, and then you let them sort of mix around and get comfortable, and then you amputate the head off of the resulting animal. What head shape will you get? And the important thing isn't the answer. The important thing is that none of the models that we currently have, all the molecular information about how stem cells turn into different cell types and all of that, we don't have any models that make a prediction about this experiment. And that's because we understand the mechanisms of cell specification pretty well, but we really don't understand the algorithms here at all. We don't understand how the collective makes decisions about what head shape will be enough to make the cells stop rebuilding, okay, and how the cells decide that a correct head shape has been formed. We really don't understand this at all. And so this kind of collective intelligence is something that I think, artificial life can really contribute. So individual cells are extremely competent. So here what you see is a single cell. This thing is called the lacrimaria. It has no brain, no nervous system, no stem cells, just one cell. And you see that it's basically handling all of its morphological, behavioral, and physiological needs at the level of a single cell. It's quite competent at what it does. We would love to have soft body robots that have this degree of dynamic control. It's hunting for bacteria in its environment right here. And so the cool thing is that these cells did not give up their intelligence when they joined into bodies, but what they did have to do is learn how to cooperate towards much larger goals. So this animal has all kinds of homeostatic, little tiny cell level homeostatic states that it's trying to maintain. But when they get together, they maintain large ones. And what I mean by that is illustrated in this experiment. So in order to become a frog, tadpoles have to rearrange their face, because tadpole faces look quite different from frog faces. So they have to move their eyes, their nostrils, their brains, their jaws. Things have to kind of move around and deform to go from tadpole to frog. And what we found was that, well, it used to be thought that what the genome somehow encoded was a set of hardwired movements that basically, since all tadpoles look the same and all frogs look the same, if you just move everything the same amount every single time, you'll get from a tadpole to a frog. So what we made was these, what we call Picasso tadpoles. These are basically tadpoles where everything is in the wrong place. The eyes are on the back of the head, the jaws are off to the side, the nostrils are up on top, everything is just all over the place. And the amazing thing is that these tadpoles largely become perfectly normal frogs. All of these organs move around in really unnatural configurations such that they will still end up eventually in a correct frog face, and then they stop. So what the genetics does not specify is a set of specific movements. It gives you a piece of hardware that executes a highly flexible program that does error reduction. What it does is continue to deform from unexpected starting conditions until the error towards a specific final outcome is minimized. And so this raises an obvious question, which is that how are these collective goals set? How does the system know what a correct frog looks like? And so what we do in our group is we've added to this kind of feed forward system, we've added these feedback loops that allow both at the genetic and at the physical level, and this is the one, the physics is what I'm going to tell you about today. They allow a progressive reduction of error. So this is basically a homeostatic loop. Anybody who has worked with a thermostat or does control theory or anything else will recognize this immediately. This is a simple homeostatic loop. But what's new about this is that the set point of this is not a single number or something simple like metabolic state or pH or something like that. The set point here is a complex anatomical state. And if the animal or plant is deviated from that anatomical state, and this could be an injury, but it could also be a mutation, or teratogens or anything else that deviates the animal from the correct large-scale plan, then a bunch of mechanisms kick in that will try to make changes to reduce that error. And so this, and this is a very non-mainstream kind of view of how this part of biology is supposed to work, suggests a novel strategy. Instead of targeting the individual genes and trying to intervene at the lowest level of genes and proteins, which is where a majority of the work today is focused, could we simply rewrite the goal state? Could we reset the set point, if in fact there is a set point, if the tissues are storing some kind of a representation of what they're supposed to be building and repairing, could we simply alter that, leave the cells alone, and let them do what they do best, which is build to that spec? And the reason this is an important strategy is because currently biology is very good at this. It's very good at figuring out what genes interact with other genes. But what we'd really like to understand is anatomy. We'd really like to understand what makes a hand different from a foot. And we'd like to, even more so, understand the dynamical goal directedness of this process, how it is that you can get from different starting positions to the same outcome, even with these perturbations along the way. And so the end game of all this, what we would like to have at the end of all our work, and we're nowhere near this, by the way, but this is, it's good to have an idea of what the goal is. The end game for all of this is a kind of anatomical compiler, that what we would like to be able to do is to sit down in front of a system that is almost like a CAD system, and we would like to draw our animal at the level of anatomy. So here it is. I'd like this worm. I'd like it to have 3 heads in the shape of this T. I want 6 eyes, and the nervous system should be shaped like this. So you want to be able to design living form at the level of anatomy and have the system compiled down into a set of low-level instructions that you would have to give to the collective to make it do this. And what we would like to do is, which is I think different from mainstream, is to really exploit this pattern homeostasis. We want to offload all that complexity onto the cells and control this whole thing with inputs or experiences or stimuli, not rewiring the cellular hardware. And the reason that's important is because look what's happened in computer science. Back in the day, in order to program, you had to rewire you had to rewire the machine. You had to interact with it at the level of hardware. And of course, I don't need to tell this audience what the incredible progress has been made by realizing that if the hardware is good enough, then you don't need to rewire it to run different kinds of software and have different kinds of outcomes. You can work at a higher level, tracking the algorithm, and you can program the thing with inputs or experiences or stimuli, not by physical rewiring. So then the question becomes, how reprogrammable is the biological hardware? Is it really reprogrammable? I'm going to try to convince you that the answer is yes. So how would we go about rewriting these anatomical goal states, the states that collectives try to implement? How would we go about rewriting them? So we had one obvious example of this, and that's the brain. And in the brain, the way things work is that the hardware is a set of cells that are connected by electrical synapses. And each cell has these little ion channels, which are these proteins that allow ions to go in and out. They set an electrical state, and that state might propagate to the neighbor. So you've got this electrical network where every cell tries to occupy some particular resting potential or electrical gradient, and then those gradients can propagate through the network, and that's the hardware. And it is thought that this hardware underlies the software that we see as behavior and cognition. So here, what you're seeing here is some voltage and calcium imaging of a zebrafish brain, a live fish, as it's thinking about various things in that assay. And there's this idea of neural decoding. The idea is that if we only knew how to interpret these electrical signals, we would know what the animal was thinking about. And there's been some progress on this, trying to infer semantic cognitive states from maps of real-time electrical activity. Well, it turns out, the amazing thing is that it turns out that all cells do this. So all cells have ion channels. Most cells have electrical synapses to their neighbors. And what you're seeing here in the bottom right is an early frog embryo, and the different colors are the output of a voltage sensitive fluorescent dye, very much like what you see in the fish above. And it's showing you all the electrical conversations that cells are having with each other. Now, what these cells are trying to figure out is who's going to be anterior, who's going to be posterior, left and right, dorsal, ventral. They're trying to sort out all of the anatomical decisions that have to be made to build an embryo. But we have the same task in our group, which is cracking the bioelectric code. We want to be able to read these electrical patterns and understand what the cells are saying to each other in terms of getting closer and closer to that anatomical set point. Now, the tools that we've developed for this, so I've shown you the voltage die. We also do a lot of computational simulation to try to understand from the expression of various channel genes, how do you get to the voltage patterns that we see here? So we try to understand these voltage patterns. And I'm going to show you two quick examples of real patterns. The one above here, and this is a time-lapse movie of a frog embryo putting its face together. And you can see there's all sorts of complex electrical activity. And this is time-lapse over about 12 or 18 hours. And this is one frame on the right, is one frame taken out of that movie. And you see something amazing. What you see here is that the bright is where the depolarized cells sit. And what you see is that the future pattern of gene expression and of the anatomy of the face is already partially laid out in this electrical pre-pattern. These electrical traces are a pre-pattern, a scaffold, an early memory of what the face is supposed to look like. And then these electrical properties guide the genes that turn on and off to then build. So you can see here off to the side, the placodes, you can see the eye here, the animal's right eye, the left one comes in later. You can see the mouth, all of these things are here. And this is a critical aspect of building the face. And now if we were to artificially change this electrical pattern, if I take these eye cells here and I hyperpolarize them and then put a depolarized eye spot somewhere else, guess where the eye will form? It will form somewhere else. And so that is how we made those Picasso tadpoles I told you about. It was by messing with this native pre-pattern. So this is an endogenous pre-pattern of guiding how the face is developing. Now, there are also pathological patterns. We're going to talk about cancer in a few minutes, but basically what you can see, here's a tumor in a tadpole that's induced by putting in an oncogene from a nasty human mutation, and it makes these tumors. But even before the tumors are anatomically apparent, you can already see from the voltage map that these cells are aberrant. They're not like their neighbors. What they've done is shut off their electrical communication, and they've basically started to go unicellular. They're starting to treat the rest of the animal as just environment. They're not in electrical connection with the whole, and this will become really important towards the end. So this has obvious diagnostic potential. But aside from being able to track and model those patterns, of course, what we really need to do is to be able to edit them on the fly. We really need to be able to read and write these electrical states. And so the way we do that, we don't apply any electrical fields. Okay, this is important to know. There's no electrodes, there's no batteries. We never apply electric fields. What we do is we go in and, guided by our computer models, we open and close the native ion channels and electrical synapses or gap junctions that control these networks. So here's a non-neural tissue in the body, and what we will do is we can either open or close these different synapses, and that means we control the topology of the network, and we can set directly using either drugs or optogenetics or misexpression of various channel genes. We can set the electrical potential of whatever cells we like. So now we have the control of cell state and the propagation of those states to other neighbors. Okay, so we have some degree of control over these electrical states. Okay, so what? Why is any of this interesting? Well, it's interesting because when you do this, you can make radical coherent changes to anatomy. So here's one example on the left. What you're seeing here is a frog embryo. Some of the cells that are going to become gut were injected with one particular ion channel RNA to set the voltage to an eye-like state, and these gut cells make an eye. Now, several things are interesting. First of all, prior to this, it was thought that only anterior neural cells up here are even competent to become eye. That's not true. You can make eye out of almost any cell group in the body. That's the first. The second thing is that this eye is really complex. If you section it, you find retina, optic nerve, all the kinds of things you would find in a real eye, but we didn't have to specify any of that. All we specified was a very simple input that says build an eye here. And so probably no surprise to this audience, it looks very much like a subroutine call. What we found is a hook into the native program that this animal uses to build an eye, and it turns out you can call that module almost anywhere. Okay, and the subroutine call for this is a spatial electrical state. Now here's another example. Here's a planarian. You amputate the head and the tail, and this middle fragment has to figure out how many heads it's supposed to have and where does the head go and where does the tail go. And what you will see here is that it has this interesting voltage gradient that says put the head right here where the depolarized or red cells are. And if we artificially manipulate this gradient and depolarize back here, then these cells, this is the pattern that the cells are consulting to know what to do. If you change the pattern, they don't know any better. This is their reference frame, and they will then happily build a two-headed worm, or in fact a no-headed worm, depending on how you change the pattern. And it's that same modularity. We don't know how to build a planarian head from scratch. We don't know how to manage all the different complexities that go inside that planarian head, but we do know the signal that now says build a head right here. It's a high level morphogenetic signal. Now in this example, what you're seeing is us triggering a second normal head. These heads are completely normal, and it's a normal head belonging to this species. And why do I make that distinction? Because actually you can call up heads belonging to other species. So you start off with a planarian with a triangular head, chop off the head. We don't touch the genome at all. So in all of these cases, the cells are completely wild type. What we do is we perturb the electrical network in a way that lowers the ability of the cells to communicate for a while. And then we wash out that drug, and the network settles back down into a different bioelectric state. And sometimes it settles back down into the same attractor that it used to have. And so it builds this nice triangular head. But sometimes, stochastically, it will settle down into states corresponding to a flathead, like a P. felina worm, or a round head, like an S. mediterranea worm. Now, these animals are about between 100 and 150 million years of evolution distant from this guy with a triangular head. And the odds of getting any one of these heads is roughly proportional to the evolutionary distance between these species. So what you can see here is that without touching the DNA at all, you can call up attractors in the state space of this bioelectric circuit that correspond to other ways of being a planarian, other species of planaria. And it's not just external head shape, it's also the shape of the brain, and it's also, this is the green thing here, and it's also the distribution of stem cells that becomes exactly like these other species. So now, okay, so we can make extra heads, we can make heads belonging to other species, but we can also explore areas of the morphospace that are not currently occupied by evolution, probably because they're not viable ecologically. So they just wouldn't survive in the real world. But you can, from perfectly normal planarian cells, you can make these spiky forms like this. You can make these cylindrical things, or you can make this that is basically a planarian with a big tube growing out of it. So these are all other regions of morphospace, and this is an example of life as it could be. And it doesn't require any changes in the DNA. It requires changes in the software, the electrical conversations that these cells are having with each other to figure out what they should be building. And you can explore this morphospace using these cells that we have. Now, one important thing to realize, and I'm not going to spend a lot of time on this, but let's not be misled to think that all we're able to do is screw up normal patterning. Okay, the point is not that we can mess up the system and get monsters. The point is that we now have pretty, at least in some of these cases, pretty good computational understanding of where the bioelectric signals come from and how to change them, so that we can now actually induce rational repair. So this is just an example of using our computational model to take tadpoles that have this really drastic defect. It's a birth defect induced by either mutations or teratogens like alcohol, nicotine, things like that. And we are able to rationally repair by designing a strategy of channel opening and closing that will actually get you from this horrible defective state to a perfectly normal tadpole brain, where they get their IQs back, they get their behavior back, and so on. Because we're able to modulate this electrical pre-pattern. So we're gaining some pretty good degree of control over this now. And so one of the important things that we've learned, and I want to talk to you about this concept of pattern memory, and the idea that these electrical circuits store memories, and they stably store patterns that are not the genetic default. So here's an example. Here is on the left here, you see a one-headed worm. And if you ask where are the head genes expressed, well, they're expressed right here at the head, like they should be. Here's another one-headed worm. Again, one head, one tail. Where are the head genes expressed? Right here at the head. Now, if we amputate the head and the tail, this fragment gives us a normal one-headed worm. this fragment is going to give us a two-headed worm. Now, why in the world would this guy give us a two-headed worm? And that would be because in the meantime, what we've done is we've altered the internal representation of what a correct worm is supposed to look like. So here you see the real pattern, the wild type pattern, has one depolarized end, one hyperpolarized end. So one head, one tail. What we've done, and you can see it's pretty messy. The technology is still relatively new. We only came up with this stuff, about 10 years ago. So we're still working out the kinks. But what you can see here is that we can alter this bioelectric pattern, and in this one-headed animal, it can store at least one of two possible memories, probably more, but we know it can store two. So this pattern memory sits there and it doesn't do anything to the anatomy. The anatomy is still one-headed. This is not a readout of what the anatomy is now. It is a counterfactual representation of what the animal is going to do in the future if it gets injured. That's pretty important. Let's think about this for a moment. This bioelectric state is not simply a readout of what the anatomy is now. It is a representation, a stored information structure that tells this collective what to build if it should get injured. Okay, and so a single body can store at least one of two ideas of what a correct worm looks like. And originally when we came up with some of these ideas of set points and so on, people are very nervous about this because molecular biologists do not like this idea of cells that are not brains having some sort of goals or having a stored representation of what's going to happen in the future. People are really much more into the emergent feed forward emergent paradigm. But what you're seeing here is that we can now literally see these goal states. We can see the set points. They're right there. And not only can you see them, you can rewrite them. So let's talk about what happens next. So here's a worm. We've amputated the head and the tail using our knowledge of the electric circuit. We've treated it with a particular drug that blocks either blocks the gap junctions or it perturbs chloride signaling, and as a result, you get this two-headed animal. Now the question becomes: you wait a few weeks. The drug, by the way, disappears. We can show that the drug disappears after about 48 hours. So you wait a few weeks, and then in plain water, with no more manipulation of any kind, we're going to chop off this ectopic head. We're going to chop off the primary head. We're going to leave just that middle fragment, that normal gut piece, and we're going to ask, what does it now regenerate? So the standard paradigm says that, okay, you've gotten rid of this crazy reprogrammed head tissue, that's gone. The genetics are untouched. The genomic sequence is still the same. So surely what must happen is that this piece is going to regenerate a normal one-headed worm. But actually, you can make models of this process whereby there's another stable attractor that corresponds to a two-headed state that perpetuates. And so when we cut this worm, and the reason I'm telling you all this is because, of course, that's exactly what happens. When you cut this two-headed worm again and again, as far as we can tell forever, it continues to regenerate as two-headed. Think of what this means. A brief change, a brief experience of that bioelectric circuit, okay, leads to a permanent alteration of the pattern to which the animal regenerates upon damage. The encoded pattern of what a correct planarian is rewritable, and once it's rewritable, it's stable. It keeps. Now, as of last year, we now know how to force it back to normal. So we can take a two-headed worm and now we can turn it back into a one-headed worm. So the reason I call these things a memory is severalfold. First, it's long-term stable, like a memory is supposed to be. It's labile, meaning it's rewritable. We finally learned how to rewrite it. It has conditional recall, like that pattern memory I showed you a minute ago. It is completely latent until the animal is injured, and then it is recalled, and then it is used. And it uses the same mechanisms, ion channels and electrical synapses, as the brain uses to maintain behavioral memory, the familiar kind of memory that you're used to. So what we're doing now is we're trying to have a real multi-scale understanding of this process, whereby we build computational models of the electrical circuit and the genetics downstream of it. We try to understand at a large scale where the attractors of that state space are and how particular experiments end up in one or another of these large scale attractor states. Okay. And so we've also toyed with models like this, where specific attractors in this state space are like memories in various kinds of artificial neural networks, where they're basically representations of specific different kinds of kinds of planarian layouts. And then these electrical states trigger different cell behaviors that build according to whatever that pattern memory is. Okay, and this may be this model may be right or it may not, but this is the kind of thing we've been working on. So one of the critical things here then is to understand what's going on with the DNA. So people usually think of DNA as the software of the cell. And what I'd like to do is propose a complementary view, which is that what the DNA gives you is the hardware. The DNA tells every cell what proteins, but specifically ion channel proteins, and of course the chemical signaling machinery, what proteins it gets to have. But once you've nailed down what the hardware looks like, this forms an excitable memory, an excitable medium with some really interesting properties. It has symmetry breaking dynamics that normally reliably break to a very specific pattern, and this is what you see in normal embryogenesis, the same thing every single time. But it also has memory properties where if you alter it, the new pattern will be kept. And so what you're seeing here is just a simulation of a set of tissues, all of which have the same ion channels. So the genetics is the same for every cell, but you see here very complex patterns that are able to form through dynamic symmetry breaking. And biochemists are already used to this in terms of Turing patterns, spontaneous Turing patterns, and things like that. But this is new in the area of bioelectrics, because we can see that this genetically determined hardware is able to house multiple different software outcomes, and that interacting at this control layer is really powerful. And so I want to end by talking about multi-scale organization and artificial life. Let's remind ourselves of two examples that I started off with. The idea that both normal and Picasso mashed up embryos both end up as normal frogs, and that that tails grafted onto the flank will become limbs. So this kind of anatomical homeostatic process, the ability to get to the correct final outcome, even with very odd starting conditions, is I think a new model for swarm cognition. And it's important to realize that all cognitive agents are made of parts. Okay, so any agent that you think is intelligent or is performing some kind of function is going to be made of something. And bodies are just swarms of cells. And we need to be very clear that the magic here is not just in the molecular mechanisms, but it's in understanding how these collectives, these swarms, deploy creative problem solving that gets you to the same goal in different circumstances. You know, if we had swarm robotics that was able to do this, we would be absolutely calling this a kind of swarm intelligence. And that's what's going on here. This is a model for not just medicine, but I think swarm cognition. And for reasons of time, I won't go through all of this, but just to say, if you're interested in that aspect, here are 3 specific papers you might take a look at where this is all digested in great detail. But we are now really trying to understand how individual cells scale up their computational world from the scale of a single cell all the way up to tissues, organs, organisms, and so on. And so we have a variety of conceptual ways of doing that. But one of the things that we think about is this thing called the struggle of the parts. And it's actually almost 100 years ago, a developmental biologist wrote this paper called The Struggle of the Parts. And it's the idea that the individual, and this is the key, sort of the main message of the whole talk, is that individual components of a biological system are themselves goal directed. So yes, the large organism itself might have behavioral goals, but all of the organs and the tissues and the cells, at every scale, there's goal directedness. And what it means is this has massive implications. What it means is that if you put an eye on the tail of a tadpole, you don't just get an eye that does nothing. It actively tries to carry out the function that it has, which is to pass on its visual information to the biggest set of nerves it can find, which in this case is the spinal cord. Here, what you're seeing on the left is actually a bunch of tissues that have been extracted from a planarian. These little worm-looking things, they're actually one organ out of a planarian. They're the pharynx. They're what the animal usually uses to eat. And you can see that liberated from the rest of the planarian, these guys are having their own life. It doesn't last very long, right? You need the whole worm to kind of live for long periods of time, but they're going off on their own. And both in this simple behavioral case and in this morphogenetic case, there is competency and goal directedness at every level. And this is critical for two reasons. Number one is that this is, I think, what is really responsible for evolvability. This multi-scale autonomy greatly smooths the evolutionary landscape. It means that if you have some sort of mutation that suddenly your eyes end up somewhere other than what they should be, you're not automatically dead. You, in fact, will probably do fine because the eye is really competent at forming anywhere and at passing its information up to the brain. This means that mutations that otherwise would have wrecked this highly complex system. I mean, that was Darwin's puzzle from the very beginning. You know, random mutations, how are they going to ever do anything good for a system that's that complex instead of just wreck it? The competency of the individual parts means that if you have a mutation that changes something, all the other stuff around it will do its darndest to still execute their correct outcomes, so that the tolerance for change and for mutation becomes massive. And it really smooths the evolutionary landscape and makes it much easier to evolve. In fact, you can start searching a much different evolutionary space if you don't have to worry about the parts. If what you're really searching is the space of reconfiguration of large modules. So this is something that that we've been working on, this evolutionary story of how individual cells become coupled into electrical networks, and that allows them to scale their goals both in time and in space to a much larger electrically coupled kind of network. Now, the danger of this thing is that sometimes you'll have defections. This process, if you have one cell that becomes electrically isolated from the rest of the body, that cell, for all intents and purposes, the boundary of the self, see, in this large multicellular system, the boundary of between the self and world, the information boundary, is encompasses the whole thing. Okay, because they're all sharing in real time. They're all sharing electrical information. It's impossible for them to lie to each other. They're literally sharing their internal contents through the gap junctions. They in effect become one creature from a computational perspective. But if that process breaks down, this cell now its radius of itself is now very small, because it is no longer plugged in or connected to this network, and now all of the signals come from outside through the cell membrane, and it has a choice. It can believe them or disbelieve this information. It can ignore it, and these cells do what unicellular organisms always do. They proliferate as much as they can, and they go wherever they want. And that is metastasis. That's cancer and metastasis. So one way to understand cancer is as a breakdown of informational connectivity that normally harnesses individual cells toward large-scale anatomical goals. Cancer cells are not more selfish than any other cell type. They're equally selfish. It's just they have smaller cells. All of these other cells are just as selfish, but they've got a giant self with a body with very large anatomical goals that they're working towards. Cancer cells have very tiny goals. And they treat the rest of the body as environment. And so you know, currently in our machines and our robotics, at least most of them, what we have is a situation where the whole machine might be intelligent and it might be doing intelligent things, but the individual parts are generally pretty dumb. You don't really have a scenario where you're worried that some of the parts are going to run off and do their own thing. And in biology, this is a constant danger. But the upside is the advantages are massive. It's huge evolvability because of this multi-scale competency that allows both evolvability and robustness. And so in cancer, we've used this, and there's just a couple of slides and then I'll stop. In cancer, we've used this strategy to normalize tumors. And we've shown that if you inject an RNA oncogene and it makes these tumors here, they're labeled in red, all you have to do is provide a bioelectrical, either a channel or gap junctions or the various different ways to artificially force these cells into communication with their neighbors. And when you do that, there's no tumor, even though the oncogene is strongly expressed, because these cells are not irrevocably broken. They're not necessarily damaged beyond repair. They don't necessarily need to be killed by toxic chemotherapy. They need to be plugged in informationally into the rest of the communication network that harnesses them towards making normal structures instead of tumors. And so just for the last bit, I want to show you something that and this is done in close collaboration with Josh Bongard's lab, and this is the work of Doug Blackiston and Sam Kriegman in Josh's lab. And so we asked this question of how much plasticity do normal cells really have? You know, we've seen in planaria, they seem to have plenty, but we wanted to do this in a vertebrate, and we wanted to see if their morphological goals were hardwired. So what we did was we took a bunch of cells, we liberated them from the constraints of a frog embryo, and we put them together in this little depression. We didn't change them genetically in any way. And we said, okay, when they reboot their multicellularity, what will they form? Okay. And what they form are these amazing motile little machines. Now, there's a couple of interesting things going on here. This is sped up a little bit. What you see here is that they can move around. They move in groups. They move in singletons. They have some interesting behaviors. This is some simulations that Sam and Josh have done to kind of simulate what they're doing in their environment. One thing to keep in mind is these are just skin. Okay, they're just skin cells. They've learned to communicate with each other to make a completely novel little creature. It looks nothing like a tadpole. It has completely different behaviors. You can see here that as by moving around these particles in its environment, it makes you know, it's kind of it has impact on its environment. They can model now. Josh's lab can model these things in silico, and we can make different shapes. And whoops. And when we make these shapes, the amazing thing about it is that if you were to sequence them, what you would read out is 100% Xenopus laevis. All you would see is frog, and you'd have no idea that that these cells were capable of getting together and forming a completely different animal. And we have one paper out already, and there's a couple more on their way that show the amazing morphological and behavioral capacities of these things that have just rebooted their multicellularity to a completely different default structure. So the conclusions then are these. Here's what I hope you remember: that computational capabilities, infotaxis, memory, and so on, are ancient. They are not just for brains. Bacterial biofilms already had most of the tricks that we associate with brains, and that this is an example of basal cognition. In fact, this is where neurons and brains came from. They came from speed optimizing these ancient properties. And what evolution does is it exploits the physics of bioelectricity to implement networks that store these patterns. These patterns serve as memories and goal states for the cellular collective. And during evolution and during embryonic development, this expands the cognitive boundary of single cells into much bigger agents that pursue anatomical level goals. And so I think there's a couple of implications of all this for the future. For robotics and AI, we can think about new non-neuromorphic approaches to machine learning. So using architectures that are completely different from the very sort of derived specialized architecture of the brain. For biomedicine, we need to exploit the intelligence of cell collectives by using stimuli, not genetic rewiring, to reset their goal encodings, and we're doing this in a number of medical contexts. And then for synthetic morphology, we now have the opportunity to create these multi-scale synthetic living machines. And some of them will be useful and they'll do useful things, but more importantly, what we can do from them is to learn to program their structure and function by cracking this morphogenetic code, by finally understanding where and how large-scale collective goals come from. And that's a major puzzle. You know, how do you discover and then reprogram goals of complex multi-scale collectives? And I think for the future of artificial life, I think these are a great set of models to think about how much divergence there can be even in standard cells, and to try to understand how to combine bottom-up emergence with top-down cognition to implement these robust goal-seeking systems, and to really understand the plastic relationship between life and mind. And so what I would like to do is thank the various people who did the work that I showed you today. Lots of people. Postdocs, students, technicians, and our various collaborators, of course, our funders, the Paul Allen Frontiers Group, and many others. And I will just close by showing you what two-headed planaria do in their spare time. This is it. The first time I showed this at a conference, somebody stood up and said, those animals can't exist. And so now I bring this video at the end to make sure that everybody can see what they look like. And I thank you very much, and I'll take questions.
Thank you so much, Mike. Yes, this is fantastic. Just a couple of logistical points about Q&A. For those that don't know, Mike is in the Eastern Standard Time Zone, so it's dinner time here. I would like to be respectful of his time. Mike, during your talk, you've accumulated probably several hours worth of questions. So what I propose we do is I'll let you field 2 questions. We've also reached the end of our hour, so there is the tutorial on molecular mechanisms starting now, molecular programming of swarms. That's starting in lecture hall A. So if you're interested in that tutorial, you can leave this lecture hall, which is lecture hall B, and join lecture hall A. Everyone else can stay on while Mike answers 2 questions. Clearly, there are many questions that Mike is not going to be able to get to. If you typed your question into chat, it will be copied and pasted into the Slack, and it'll be pasted into the hashtag keynotes channel. So if Mike doesn't get to your question, and Mike, if we can impose on your time some more, you don't have to do that this evening, but sometime during the rest of the conference, if you want to dip in there,
Sure.
You can interact with those that have more questions for you.
Absolutely.
There will be other keynotes that are dropping in there. So if you have a question for Mike, drop it into hashtag keynotes and address it to @Michael_Levin. Okay, I believe that Hiroki had the first question. So Hiroki, you had an involved question, so I'll let you answer it. So Hiroki, if you're still here, please unmute your microphone and ask your question.
Yes, okay. So thank you very much, Mike. So this is a great talk. And my question I posted, almost 20 minutes ago was, do you have any illustrative cases where this amazing biological adaptivity plasticity actually fails miserably? So
Sure.
we want to hear more about that kind of stuff.
Sure. Yeah. Oh, yeah, absolutely. And this is where all of these systems have limitations, and the limitations are seen clearly in cases of birth defects, and they're seen clearly in cancer. So so I'll take the birth defects case first. So depending on the species and the kind of birth defect that we're talking about, there is anywhere between 1% to 10% of embryos at particular stages that are simply incorrect. And there are various ways to break the ability of the system to repair back to normal. So the way this happens is it can be caused by problems in the environment. So either chemicals or temperature or various other things in the environment that break the ability of the system to get back to normal. It is sometimes caused by other animals. So for example, bacteria or other living things. So bacteria, for example, certain kinds of bacteria can cause planaria to develop either 2 heads or defects of the visual system, because they've evolved to plug into the same electrical signals that the animal is normally using. They're basically hijacking that system. So parasites of various types can hijack that system. And then of course there are genetic mutations. So there are channelopathies. There are mutations in ion channels that render the system unable to form the patterns that normally form. And those are the things that we try to fix. These are the targets of our repair. So absolutely the system can be overwhelmed. And cancer, same deal with cancer. So normally the cells are electrically normalizing each other to keep everybody away from cancer, but sometimes that fails, and that's when you get cancer. So for sure, these systems break.
Yeah, thank you very much. Very interesting.
Okay, the second question, and I apologize if I mispronounce your name, Abdalrahman Mustafa. If Mr. Mustafa is here, please unmute yourself and ask your question. If not, we will move on.
Yeah, I'm here. All right. So, well, thank you for the great lecture. This was like incredibly inspiring.
Thank you.
So yeah, my question is, what do you think, even if that's just guessing these problems, what do you think about the embryological problems or the congenital problems that involve not finding a genetic anomaly to do with them? So like people do genetic wide association studies and they simply don't find patterns. What do you think about those? Can these be explained by looking at how biology works? Thank you.
Yeah, I didn't get all of that. I think the network wasn't great for part of that. What I heard was how does all this relate to the kind of genetic screens that people are doing? Is that about right?
I can read the original question very quickly if that
Please.
helps. So I'll do embryological malformations with recognized genetic errors happen. This
I
is
see.
really interesting. What about malformations for genetic errors, idiopathic anomalies?
I see. Okay. So what happens is, right, so there's a couple of different ways. I mean, the easiest way to think about this is if you wreck your hardware to a sufficient degree, then no matter how good your software, you're not going to be able to carry out certain functions. So there are mutations in the genome. If you need a particular enzyme to turn one chemical into another chemical, and you've had a mutation in that enzyme and it doesn't work, there's nothing that bioelectric signaling is going to be able to do. There are tons of these kinds of mutations. There are other mutations in ion channels which themselves set up incorrect bioelectric patterns, and then you get defects. There are yet other mutations that we can fix. So for example, we've shown that there's a gene called Notch, which is a very important gene for brain development. We take embryos that have a terrible mutation in that gene. By forcing the appropriate bioelectric state, they will still have pretty normal brains. So you can often intervene despite the fact that you have some of these. And it just depends on what case you're talking about, and what kind of damage it was that you've had.
Okay, I think we're doing pretty well here. So if we can impose on your time a little bit more, Mike, maybe we'll allow one last question.
Sure.
Stuart Bartlett, if you wanted to ask your question.
Hi, thank you very much, Professor Levin. It's absolutely a phenomenal talk. I really enjoyed it. And I wanted to ask about whether we need to, well, almost certainly we need to modify our definition of set points, because there are clearly either attractors or goals that these systems are heading towards. But as you showed, they're so plastic and they're so dynamic, and there's a lot of switching and decision-making involved in the definition of those set points. And I just wondered whether you think we need to somehow change our definition, or whether it should be sort of merged into a more general conception of biological learning.
Yeah, so there are lots of potential subtleties. There's an incredible amount of room now for development and for new work, and there are lots of subtleties that I completely glossed over for two reasons. I mean, there's allostasis, which is distinct from homeostasis. There's a million different people who work on this stuff, really, in cybernetics and computer science and so on, may say this is an incredibly kind of that one loop I drew for you is very simplistic, and it is. And the reason is twofold. First, just for lack of time, we could have a whole deep discussion about it. But the bigger reason is that whole notion in itself of a set point that is an anatomical state, and this idea that the embryo is actually trying to get to a particular outcome, is basically a forbidden territory in molecular and cell biology, right? So biology has been running headlong away from anything that sounds like teleology for many years, and people are really uncomfortable with that idea. Now, I point out for those audiences, I point out that people in control theory, engineering, cybernetics have already had a non-magical theory of goal directedness for decades. So I don't think we have to, worry about it all that much, but it's still a really controversial concept in with my colleagues. And so for this reason, at that point, I'm just showing you the very basic, simple, I'm trying to demonstrate a really simple fact that these goal states exist, and that we can see them and we can rewrite them. And having done that, I would love to have more in-depth conversations with people who actually work on this stuff to nail down some very specific models of what this really is, and the determination of those goals and the representation of those patterns distributed along cell networks. I mean, these are all things we should be discussing.
Excellent, thank you.
Thanks.
Okay, I think this is a good place to pause. I will invite everyone in the audience to move the party to hashtag keynotes. And Mike, you're welcome to carry on over there and continue answering questions now, or join us again tomorrow and later in the conference and keep the conversation going.
Cool,
So
thanks so much.
Again, Thank you again, Mike. And just a reminder that we are starting up again tomorrow morning, and at 9 a.m. Eastern time, we have our second keynote speaker, Sarah Amari Walker from Arizona State University. So thanks all for a great first day. Thank you, Mike, and we'll see you all tomorrow.
Thank you, everybody. Bye.